So we are going to refer about what is resting potential. Okay. Then in resting potential, I have brought the chart that is relative concentration of sodium ions, potassium ions, chloride ions inside the plasma membrane and outside the plasma membrane, and the ratio of the ions. Okay. So before that, we will be referring what is the basis? Why we have to refer the basis? That is the charge on the membrane. That is because of the ions, and how the propagation of nerve impulse is conducted across the neuron or the nerves. Hence, we are going to refer first about the concept of resting potential. So, potential is actually we can refer it in the terms in the context of charge or voltage, and it is because of the positive and negative charge on the ions or molecules across the plasma membrane of the neuron. So now in this component of resting membrane potential, we should be aware of different other definitions of that will be referring in due course, such as depolarization, polarization, repolarization, and also the hyperpolarization in the context. Okay, so we will be referring in the diagram from dendrites to the axon, how the propagation of nerve impulse is occurring. And just I mentioned here, yesterday I was referring about the myelinated sheath. Okay, so how myelin sheath is formed? That is the glial cells. Okay, the ultrastructure, because the neuron is there, and about that is surrounding the neuron, these glial cells in the central nervous system and peripheral nervous system, they form the myelin sheath. And there is a difference between the myelin sheath. Ultrastructural difference is that is found in the peripheral nervous system, in the axons of the peripheral nervous system, and those in the central nervous system. Okay, so now let us refer here and also about the ion channels. So you should be referring about ion channels. There are ion channels are then sodium channels, potassium channels, then sodium potassium transporters. Okay, and the gating and the closing and opening of these channels is important. And what is the function of means the propagation of nerve impulse? The potassium ions, chloride ions, and the sodium ions. Okay, so I will write here the basis. That's why I mentioned previously to refer the action, the resting potential. So what is resting and what is action? Why it is called action? Okay, or that may be referred to the term firing of neurons. Okay, so what is that? We will be referring now. So that's why the basis. So you should this question may come with the example. What is the resting membrane potential? What is the concentration gradient? Okay, so you should be accordingly take the answer. Okay. So the basis of membrane resting membrane potential. Resting membrane potential. I will be drawing the diagrams also here of the various ion channels. They are the proteins actually, and their function is to allow the entry and exit of certain ions such as potassium, sodium, and chloride ions. Okay. So now let us start. For the neuron system to function, the neurons must be able to stay and receive the messages or the signals. Okay. So that is the importance. That is how we refer the functioning of the nervous system, because it's a nerve cell. We have referred the structure, and they should be able to receive and transmit the impulses to the neighboring or other neuron or to the target or effector organs in the context, as they are according whether they are sensory or effector or motor neurons in the context. So these signals are possible because each neuron has a charged cell membrane. So charged cell membrane. So usually it is negative inside and positive outside the plasma membrane. Minus 70 millivolts is the current when it is. Just now somebody asked me Yes, one of those you asked me about the practical. So in the books there is an experiment is given. Practical, because I am referring to the context of the experiment. So electrode is dipped into the plasma membrane of squids. They are the mollusks, and they have recorded there is a voltage, and that is, for example, it is minus 70 millivolts. Of course, although it differs from species to species, and also depending upon the neurotransmitters, and also the depending upon the sensory, that is actually another component, but it depends on the species and the that is the membrane potential in the context. So, a voltage difference between the outside of the plasma membrane and inside is called the membrane difference. So, here we are referring in the context of the ions and the charge on the cations, anions should be referring and properly mentioning the answer. This question may be there, can be asked. Usually, question next topic of this after this is action potential. So to answer the question of action potential in the myelinated and non-myelinated neurons, you should be first aware about what is the resting potential. Resting means when the neuron is at rest in the context. Means when there is no conductance of impulse across the neuron, that context is referred as the resting membrane potential. Means it is not depolarized in that context. So the change of this membrane can change in response to the neurotransmitter molecules released from the neuron and also the environmental stimuli. So depending upon the stimulus, as we referred to, the signals for the sensory neurons and the neurotransmitters, there are various types of neurotransmitters such as acetylcholine, histamine, even dopamine, I referred to. Various types of neurotransmitters are there. And according to the release of neurotransmitters, there is a change in the action of resting membrane potential. So to understand the neurons, how neurons communicate, one must first understand the basis of that is the resting membrane potential. That is the basis of the resting membrane potential. That is means now here you can, for example, correlate with other types of cells. You may get a concept as to what is the epidermal cells, muscle cells, other types of epithelial cells or fibroblastic cells. So whether it is similar to the neurons, yes, that is a difference, because there also it may be more of negative inside than plenty of the plasma membrane of the outside. And although those cells don't contain impulses and neuron cells they contain impulses, hence they are cells they are the basic and fundamental structural unit of the process. So now neuronal charge membranes. So here I want to, since you are having a topic, I will first write the chart. I draw the chart here. There is also a diagram. Okay, so there is a table. This table is important. You can also write this table. Ion concentration. Ion concentration. Inside and outside neurons. Inside and outside neurons. So the ion concentration. So whether it is same, why we are writing? What is the importance? Okay, so for example, now here ion. Then extracellular concentration. Extracellular concentration. Then intracellular concentration and the ratio. intracellular concentration and ratio. So, for example, let us consider sodium ion. Okay, sodium is a positively charged sodium, and its extracellular concentration is 145 millimolar. So, it is in millimolar concentration. So, it is 145. That is, they are higher in concentration outside the plasma membrane. Across the plasma membrane, for example, this is the plasma membrane, this is the cytosol, and here there is sodium ions are more, and this is the cytosol or cytoplasm, and here the potassium ions are more. Then intracellular concentration is 12 millimolar of sodium. Intracellular concentration of sodium is 12 millimolar and the ratio is 12. Now we will check the concentration for potassium. So potassium आपण check करू. The potassium intracellular just 4 millimolar. So extracellular is just 4 millimolar. And they are less in concentration outside the plasma, potassium ions, compared to the sodium ions. And their concentration is more inside the cell, inside the plasma membrane, and that is 155 millimolar. That is of the potassium ion, and the ratio is 0.026. 0.026. Now we will refer about the fluoride ion. So fluoride ion चा संदर्भात आपण चेक केला तर it is also extracellular. It is higher concentration of fluoride ions. That is extracellular. And intracellular it is 4. कारण याच्यामध्ये जे निगेटिव्हली चार्ज प्रोटीन्स असतात फ्लोराईड संदर्भात ते रिपेल करतात आणि म्हणून फ्लोराईड आयन्स ते प्लाझ्मा मेम्ब्रेनच्या बाहेर असतात, सेलच्या बाहेर. आणि रेशो इज सिम्पल. तर हा चार्ट तुम्ही लिहा बिकॉज दिस इज द बेसिस टू अंडरस्टँड ऑर राईट टू फर्दर रिगार्डिंग द ऍक्शन पोटेन्शियल. आपण जेव्हा पुढे फर्दर ऍक्शन पोटेन्शियलचं रिपोर्ट करणार आहोत तेव्हा त्यामध्ये तुम्हाला या चार्टचं उपलब्ध आहे आणि refer as a reference so that you can get to refer the concentration, relative concentration of sodium, potassium and fluoride ions across the plasma membrane. So you can then refer accordingly. This table. आता याच्यामध्ये डायग्राम आहे तो त्या डायग्राम मध्ये